All right, hello everyone. So this is our exam review for exam 3. So first off, before we start getting into the material, I have a few announcements. So before we get started, I'm going to ask that you sign in. So the way that we'll do this, if you're not familiar with SI, we have QR codes. So if you have an Apple phone, some Androids do this, but I'm not sure all of them do. You can scan the QR code when you're in your camera feature, and it will take you to a link where you can sign in for today. So I'm going to put that QR code up now, and in a second, and I'll ask that you sign in. And if you can't get it when I put it up, I've also uploaded the QR code onto Blackboard. It's under the SI section. So if you wouldn't mind to go ahead and sign in, it's really important so that I know who's using these materials, or I know that it's helpful and that people are using them. And also that I can continue to provide these kinds of things for you. So without further ado, here is the QR code. So if you can, go ahead and scan that, and it'll prompt you to put in your student ID or your student ID number, and you'll click exam review session. So, all right, thank you. And if you weren't able to get it then, I have this sheet uploaded on Blackboard, so you can go ahead and sign in using that. Okay, also, so all the supplemental material has been uploaded to Blackboard at this point. So the exam 3 outline, which I'll be going over during this session, is now uploaded on Blackboard. Also, all of the worksheets that I created for the content that will be covered on this exam is in Blackboard at this time. If you work through the worksheets and then send me an email of pictures of the worksheets so that I can see that you tried them, then I can send you the keys. So I ask that you do that tonight or tomorrow anytime, and I can send those to you. Okay, and so also I'm going to be going through the outline. If you have any questions about anything I talked about in the outline, if something didn't quite make sense, if there are some things that I didn't explicitly mention in the outline that you have questions about, I will be holding an exam question and answer session tomorrow in Davidson 209A from 5 to 5:50 p.m. So again, that session will be tomorrow in Davidson 209A from 5 to 5:50 p.m. I'll be sending that out in the email. So if you have any questions, further questions before the exam, please feel free to drop by or to send me an email. That's fine too. So without further ado, we're going to go ahead and jump into the material. So first off, also I have the outline here. This is why I'm looking to the side. So we're going to start off with our first lecture, which is complex inheritance, sex determination, and non-Mendelian genetics. Okay, so my first point is, what are the 2 factors that determine biological sex? And so those factors are your sex hormones, which are produced by the gonads, right? And then our sex chromosomes. So first, on the topic of sex hormones, we want to think about what are the sex hormones? Well, those would be your androgens and your estrogen for female. So your androgens for male characteristics, your estrogens for female characteristics. And when you think about these sex hormones, think about them promoting sexual characteristics for each biological sex. For example, androgens are also associated with testosterone, which is females have some testosterone, but males have more testosterone. And those are secreted, those target the gonads, the male gonads, which are the testes. And for females, the predominant sex hormone is estrogen, and these are associated with the ovaries. So again, it's just hitting home the importance of the production, the proper production, proper reception of sex hormones, and also the presence of sex chromosomes. And so if we want to think about sex chromosomes, we want to think about what are the sex chromosomes that are present for females and for males. So for a male, we're going to have 1 X chromosome and 1 Y chromosome. And for females, that's going to be 2 X chromosomes, okay, which is important to note. Also, that kind of ties into the next point, how many pairs of chromosomes are there? And within that, how many are the autosomes and how many are the sex chromosomes? Well, so for the first question, humans have 23 pairs of chromosomes, and that's all inclusive. However, we can break those chromosomes into two categories, and that's the autosomes and the sex chromosomes. Now, the autosomes are comprised of 22 pairs of chromosomes, and the sex chromosomes are the 23rd pair of chromosomes, or the last pair of chromosomes. Okay, so again, the autosomes are pretty much all the chromosomes that are not sex chromosomes, and that is 22 pairs of those, and then you have one pair of your sex chromosomes, and those, of course, determine your sex. So if you have two X sex chromosomes, then you're a female. Or an X and a Y chromosome, then a male. And of course, there are some abnormalities that we will get into in a moment. Okay. So next, what is the name of the gene on the Y chromosome that promotes the development of male sexual characteristics? And that is your SRY gene. Okay. Again, SRY gene. It's important to note that the Y chromosome is very small. It doesn't carry that many genes, that much DNA. It predominantly carries genes that promote male sexual characteristics. So that's why we don't really see the Y chromosome undergoing recombination or anything like that, because it is so small and does not carry that many genes. Okay? All right, so what causes ambiguous sex organs? There are several things that could cause this. Number one, chromosomal abnormalities, which I kind of alluded to earlier. So for example, with Turner syndrome, okay? So Turner syndrome is when you have an X chromosome. Oh, that's backwards. All right. That's when you have an X chromosome and just the one. You don't have two X chromosomes that would be normally associated with a female. However, an individual with Turner syndrome would still be biologically female. However, there would be possible infertility, broad shoulders, webbed necks. There are other things associated with it. However, chromosomal abnormalities like this can be present. Also, for another example, we have XXY, which is for Klinefelter syndrome. And that is when you have a male who has an extra X chromosome. And that can also cause some infertility issues, things like that. There is a chart in your lecture slides that goes through some more of these chromosomal abnormalities, but those are kind of the two big ones that I wanted to mention to you. Also, ambiguous sex organs can be caused by the no production of sex hormones at all. And of course, this brings to light the importance of sex chromosomes with promoting sexual characteristics in males and females. For males, the androgens, for example, testosterone. For females, estrogen. And so if we're not producing these sex chromosomes, we have issues promoting our sexual characteristics. So that's a very important thing to note. Also, there could be issues with there being no receptors for the hormones. So in this case, it would be someone who produces sex chromosomes, for the sex hormones, excuse me, but they don't produce the receptors to be able to recognize those hormones. So in both cases, you're not able to promote sexual characteristics to the extent of a normal individual. And normal in the sense that they have the necessary receptors and hormones. So all of these can cause issues or can lead to ambiguous sexual organs. Okay? Okay, next, what is an X-linked trait? Okay, so what we've talked about so far and we talked about for the previous exam was alleles. So for example, if we were going to talk about height, and we wanted to say that big H is dominant for height. Well, let's do hair length. So we'll say that big H is dominant, and that is for long hair. And we'll say that little h is recessive, and it's for short hair. And so for this example, we're not talking about the sex chromosomes carrying the trait. We're talking about alleles for genes on chromosomes that are not the sex chromosomes, the autosomes, right? But when we're talking about X-linked inheritance, we're talking about traits that are carried on the X chromosome, the sex chromosome. Okay, for example, red-green color blindness. So red-green color blindness is a recessive trait that is passed on the X chromosome. So, for example, if I was to show a male who is red-green colorblind, then it would look something like this, where the little r, and I'm sorry, it's a little bit hard to see, where the little r on the Y is showing that let me get another color because I'm still not happy with that. Okay, so I will use the red color to denote that the X is affected, okay? So this X is carrying the gene for red-green color blindness. Now, my question to you, number one, for this one, what is the sex of this organism, of this individual? XX. Hopefully you said female, right? Because 2 X's indicates that we're female. And if one of the X's is carrying the red-green color blindness gene, but the other is not, will this female express red-green color blindness? Will that be her phenotype? And hopefully you said no, right? Because if red-green color blindness is recessive, that would mean that we need 2 alleles carrying that trait in order to express it. Okay, so a female who has one trait, one X chromosome carrying the trait for red-green color blindness, and one that is not, one that's normal, right? Then this female is not expressing red-green color blindness, but she is a carrier, okay? And that becomes important. So let's see what happens if a female, a mother who is a carrier for red-green color blindness, has children with a male who does not have red-green color blindness, okay? So let's just go ahead for old time's sake and set up a good old Punnett square. Okay, so as we can see, I have drawn in a female who's a carrier for red-green color blindness and a male who has normal vision, okay? And so now I'm just going to go through and fill in this Punnett square. So as you can see, the X, right? So we're kind of just putting this together. If you have any problems with doing Punnett squares, I have some videos that I've linked in the bottom of the outline that can help you, but also you can come to the help session and that can help explain Punnett squares. Okay, so I filled in my Punnett square, and now we can pretty clearly see that as far as my females are concerned, right, which are my only individuals with only X chromosomes, my females, so I have one female who has completely normal vision, but I have one female who is a carrier. Now, she does not express red-green color blindness, but she's a carrier, so then her children could have red-green color blindness, okay? Now looking at my males, it's kind of a different story. So one male has completely normal vision, not a carrier or anything. And one of the males expresses red-green colorblindness. Why? Because there's only one X in males. Therefore, if a male inherits an X-linked chromosome that carries the trait, then they will express it because they only have one X. For females, that isn't the case because there are two X's. And so in order for a female to be red-green colorblind, she would need to be homozygous for the trait. So she would need two diseased or two X's carrying the red-green colorblindness trait, okay? Now, my question to you is, could a male be a carrier for an X-linked trait? So hopefully you said no, because as we just saw with the Punnett square, for a male, if the male has an X that is carrying a trait for an X-linked, carrying an X-linked trait, then they will express that because there's only one X present. Okay, so that's something that's important to note. The rules for recessive inheritance still apply. However, it gets a little bit different when you think about how many X's there are in males and females, and that can help to determine whether the phenotype will be expressed in that individual. Okay? All right, so next up, let's talk about pedigrees. So that really plays very well into what we were just talking about. So what is the purpose of a pedigree? Well, a pedigree allows you to see the pattern of inheritance of a trait through a family, okay? And so you can see what people had a disease and how many of their progeny and their progeny's progeny carried these traits, whether they were carriers or whether they expressed it, okay? And so by looking at a pedigree, we can kind of see and fit together the pieces of what must have been that pattern of inheritance. So essentially, we can see if a trait was X-linked, if it was dominant or recessive. And so I've included some videos in here in the outline to help you with this. But for now, we can come up with an example to kind of see this. So for your purposes, it's important to know that a box, okay, is a male, and a circle is a female, okay? Whenever a box is completely shaded in, that means that that individual is expressing a trait. If the box or the circle is halfway filled in, it means that that person is a carrier. So they may or may not be expressing the trait, but if they're a carrier, they're not expressing the trait, but they still have the allele, okay? So let's think about what a pedigree would look like for red-green color blindness, okay? So let's say that we have a male and a female. And let's say that they have 3 children. Okay? Let me get that a little closer so you can see. Okay, and let's say that my female is a carrier for red-green color blindness. I'll try to make it a little bit more obvious. That is not filled in. Okay, so she's a carrier for red-green color blindness. Okay, and so when we were looking at doing the Punnett square, it's a 50-50 chance that her son will express that trait. She has 2 sons, so let's say that one of them expresses the trait. And the other son has perfect color vision. Okay, and let's say that the female got lucky and she did not express that trait, okay? Now let's say this male who has red-green color blindness has 2 children. He has a son and a daughter. Let's think about what this would look like for the next generation. So my male, who is XY, is carrying a trait for red-green color blindness, okay? The female does not have red, green color blindness. She has perfect vision and she is not a carrier. So what does the male pass to each child? So we know that this male will pass on a Y to his son, and the Y doesn't carry the trait for red, green color blindness. So and the mother doesn't have any alleles for red, green color blindness, so her son will not have red, green color blindness. However, since the father will pass on his X to his daughter, then the daughter will be a carrier. Okay? The big takeaway point that we want to make with this is that certain patterns of inheritance have certain patterns. Okay, so for X-linked, it's usually seen that the males will express the trait more often than females. And that's the case because males only have one X. Therefore, if that one X is carrying a trait, then that trait will be expressed. For females, both X's need to be expressed. Both X's need to carry the trait in order to be expressed. In the case that we're talking about a recessive trait, okay? So for X-linked recessive, males will more often express the trait, and for autosomal dominant traits, you'll just see more individuals that carry the trait. Versus for recessive, there will be slightly less people who carry the trait. So really the important part is to be able to recognize the patterns. However, we didn't talk about that a whole lot in class, so I won't spend too much time on it. We'll go ahead and move on to the next point. So if you have any questions about the pedigrees or X-linked inheritance, shoot me an email or come by the help session tomorrow at 5 for that. Okay, so we already talked about the Y chromosome, so I will skip over that one for the sake of time. Okay, so what is the difference between incomplete dominance and codominance? So this is typically two vocabulary words that get mixed up pretty often or can seem pretty similar. I remember when I was learning this, it took me a minute to realize. However, so incomplete dominance and codominance are both talking about the heterozygous individual. We're talking about the phenotype specifically of the heterozygous individual. So hopefully off the bat, you remember what I'm talking about when I say heterozygous. So if we have alleles, for example, I'll do red and pink, or I'm sorry, red and white. So let's say that I have an allele for white, okay, and red. And I'll just make that red so that you can see it easier. Okay, so when we're talking about incomplete dominance, codominance, we're really talking about the heterozygous individual. In these cases, in what we've looked at before, there's been a clear dominant and recessive allele. For example, we're saying that the dominant allele masks the recessive allele. Okay, but what if that wasn't necessarily the case? What if both the dominant and the recessive allele were actually both dominant? And saying, what if we really don't have recessive alleles? We just have alleles, okay? This is what we're talking about. So of course, if we had a homozygous for R, right? If we had this, big R, what would my phenotype be? It would be red, right? Conversely, what if I had two W's? Then my individual would be white. Okay? But now our question is, since neither of them are completely dominant over the other, what happens if I have a heterozygous individual? And that's where we talk about codominance and incomplete dominance. So for incomplete dominance, think about a mixing of the phenotype. So incomplete dominance, mixing. When you see this, you think, neither of them are dominant over the other. We're seeing an intermediate of the two. So for example, for R and W, for red and white, we would see a pink individual. We're getting a mixing of both the phenotypes from each allele. It's to say that incomplete dominance is showing an intermediate phenotype. And this is different from codominance. Now, codominance is saying that, think about you're on a sport and you're a co-captain with your best friend. Neither of you are captain of the team more than the other. Both to say that both of you are equally captains of that team. In this case, both of the alleles are equally captains of the phenotype. They're both expressed equally and distinctly. In incomplete dominance, we had a mixing, an intermediate phenotype. But for codominance, both of the phenotypes are expressed equally, both of them with their integrity intact. So for codominance, red and white would be like red and white polka dots. They're both equally present, but they're both distinguishable. You see red and white. Whereas for incomplete dominance, you don't see red or white, you see pink, you see the middle, okay? So those are the big differences between incomplete dominance and codominance. Now let's kind of think about polygenic traits. Now polygenic traits, what we've been talking about before is one gene determining one trait. For example, my example was for hair height. Big H was long hair and little h was small hair. It was to say as if that one gene would determine that phenotype. But in all actuality, in not all cases, but for some, more than one gene will affect one trait, okay? And that's polygenic inheritance. So if you've been to my SI sessions, you already know, but one thing that I like to do when I'm faced with words that I'm not very familiar with is to break that word down into parts and to think about what each part of that word means. Polygenic inheritance, poly meaning many, genic meaning genes. So many gene inheritance. So literally we're saying that multiple genes are contributing to my overall phenotype. So for example, your example is in class is height. So in all actuality, the actual height of a person is determined by many genes interacting with each other. So in this case, height would be considered a polygenic trait. Okay? So what's next? What about multifactorial inheritance? So this is to say that humans, that we do not live in a vacuum, okay? It's not just our genes involved. We have many other things going on, right? We have our environment. We have pollution or the lack thereof. We have many things that affect our inheritance. And so multifactorial inheritance is taking into play all those external factors that actually do play a role in our growth. And for example, and this is kind of a crude example, but let's say that someone had the gene structure for the genes or the alleles for being tall. However, this person took drugs as a young person or middle-aged person, and it stunted their growth. So their overall phenotype was actually affected by things other than their genes. So think about that in the way that other factors, other external factors can play a role in determining the phenotype of an individual, okay? All right, so next, non-disjunction. What is non-disjunction and what can this lead to? So non-disjunction is the failure of, wrongful splitting of the chromosome during cell division, or incomplete separation of the chromosomes. So if you remember during the cell cycle, right, during anaphase, we would pull the chromosomes to separate poles of the cell. But what happens if an extra chromosome or extra homologous chromosome was pulled to one pole than the other? To say that one pole or one soon-to-be daughter cell received an extra chromosome while the other soon-to-be daughter cell was lacking a chromosome. This is called non-disjunction. Okay, so non-disjunction is when the chromosomes fail to separate properly during cell division, and that can lead to one daughter cell having more chromosomes than necessary and another chromosome having not enough. Okay? And so this can lead to aneuploidy, and aneuploidy is just having an abnormal number of chromosomes. Okay? All right, so let's move on to lecture 12. And lecture 12 is talking about natural selection and adaptation. So this is a really cool, interesting part of biology. It's definitely one that you can see happening right outside, right here. So that's pretty cool. So first we're going to discuss antibiotics. And so what are antibiotics and how do they perform their function? And you probably already know what antibiotics are, right? You've probably, maybe not if you're, really healthy, but you may have had to use antibiotics at some point in your life. And these antibiotics are chemicals that kill bacteria, right? So if you have a bacterial infection, if you're sick, then you'll probably be prescribed an antibiotic. Okay, and so how do these antibiotics destroy bacterial cells? Well, usually these will interfere with the functioning of the bacterial cell. Specifically, there are some antibiotics that can inhibit cell walls or bacterial cell walls. And so if the integrity of the cell wall is compromised, then the bacterial cell is then more susceptible to that antibiotic, more susceptible to the environment, and they are not able to protect themselves. They're destroyed. Okay? And what about binary fission? So what is binary fission? Binary fission is reproduction of bacterial cells. Okay, and when we think about this, we want to think about what's the difference between sexual and asexual reproduction. For sexual reproduction, we have a mixing of DNA from two individuals, right? So, for example, well, you don't need an example of sexual reproduction, but sexual reproduction is two different organisms, two different individuals combining their DNA, mixing their DNA to make offspring. And now it's important to note, very important to note, that the resulting offspring of sexual reproduction, the resulting offspring of sexual reproduction will be genetically different from each other and from their parents. Okay, so this directly contrasts asexual reproduction. Now, asexual reproduction only involves one individual. Okay, so one individual splitting into two. Now, you're probably thinking, well, we're not mixing our DNA, so how are we genetically different? We're not, right? So binary fission is asexual reproduction, just the splitting, literally, like this is one bacteria and we're splitting, oh, we're two. That's binary fission. Okay, and each of those bacteria, those resulting bacteria, are genetically similar. They're genetically identical to each other and genetically identical to their parent cell. Okay? Okay, but it's important to note that there are ways for bacterial cells to acquire new DNA. And this can be through mutations, right? And even we can get new genes, right? New traits through mutations. So bacteria cells are not immune to this. They can gain new genes through that mode as well. But also we have gene transfer. So there are ways for the DNA of other organisms, other bacterial cells to be absorbed or taken up by that bacteria, but we won't go into specifics of that. It's just important to know that there are ways for the bacterial cell to be able to get new traits, new DNA, but we won't go into the specifics now. Okay. So next off, what is a population? So a population is a group of organisms of the same species who live together in the same geographic area. Okay, and this becomes important when we're talking about population ecology and things like that. But right now, it's just important to know that a population is a group of the same species living in the same geographical area. Okay, and so bouncing off of that, what evolves? Can individuals evolve? Can populations evolve? Well, individuals cannot evolve. Populations evolve. So the process of evolution, as we'll talk about even more later, is driven by natural selection. And natural selection is saying that each population has a different frequency of alleles or traits in their population. Okay, and so the frequency of these alleles can be altered depending on what traits are favorable for a specific environment. And of course, that could stay the same or that could change. We see the change in allele frequencies in a group of individuals, in a population. But looking at just one specific individual will not show the process of evolution. Okay, we need to see the change in frequency of a large group of populations to be able to see evolution occurring. Okay, so that's a very important thing to note. Individuals cannot evolve, populations can evolve. Okay, next up, what is fitness? Okay, so fitness is an organism's ability to survive and reproduce in their particular environment. So when we're talking about an individual's fitness, we're thinking about how well they're able to thrive in their environment, especially compared to other individuals. So, for example, and this is, I don't know why this comes to mind, but let's say we have zebras. Let's say that there's a genetic abnormality where there are some zebras who are very fast, and there are some zebras who are a little bit on the slower side. Now, which of these zebras do you think will be able to survive and reproduce in their environment? Well, probably the zebra that's very fast, right? They'll be able to run away from their predators. So in this context, we would say that a zebra who is able to run faster will have a better fitness than a zebra who is slower. Okay, so when we're looking at fitness, we're looking at the ability of an individual to thrive in their environment and to reproduce and pass on those traits. Okay? Okay, and so that really ties in well to the idea of natural selection. And natural selection is saying that there is differential survival and reproduction rates for individuals within a population in response to their environmental pressures. Okay, so this really ties home the idea that depending on individuals with certain traits, certain characteristics are more well fit in their environment, and due to these traits, certain individuals will be able to survive better even despite environmental pressures. So these environmental pressures, or you could say nature, is selecting for individuals with certain traits. Okay, so those individuals with traits that allow them to thrive in their environment will pass those traits down to their offspring. Therefore, over time, of course, changing the frequency of that trait in their population. Okay, and that is evolution. We're seeing the change in the frequency of alleles, the change in the frequency of traits in a population over time. Okay. Okay. Now let's kind of think about the patterns of natural selection. Okay, and there are three that we want to think about. So first, let's talk about directional. Okay, so let me see this. All right, so let's say this is my curve, okay? And due to pressures in the environment, nature is selecting for individuals of the extreme phenotype. So let's say that this is black, middle is tan, and my other extreme is white. Sorry, those are backwards, I think, on there. But anyway, so my extreme to the left, or to my left, is black. The middle is tan, and the other side is white. Okay? And so for directional selection, we're saying that we are selecting for one of the extreme phenotypes. So for example, I would be selecting for my black. Or conversely, I could have this going the other way where I have more white individuals than any other. So that would look more like this. Okay? So that is directional. And now let's think about stabilizing. So stabilizing is where the intermediate phenotype is favored. So in this example, the tan phenotype would be favored over the others, the extremes, okay? And our last one is diversifying, okay? And this one is where both of my, and in this one, both of my extremes are favored over the intermediate, where you can see we have both of my humps at both of the extremes and a plummet in the amount of intermediate phenotyped individuals, okay? Again, there's a great picture of this in your lecture notes. If you have any questions still about these, let me know. Shoot me an email, come to the help session, and we'll be able to talk through it. Okay, awesome. So we are through that first lecture. And so now, or those first and second lecture, so now let's get into the history of evolutionary theory. So the great thing about this, and of course I'm a biology nerd, but this is pretty interesting stuff. Also, it kind of, when you think about it, it kind of makes sense, right? So my first point is, what was the initial belief about the change of species, or how species were created, how organisms were created? And the first widely accepted belief was in creationism, or that, each animal, each organism was specially created by God and perfectly, as to say that none of the individuals have changed over time. They're exactly the same as they were when they were created. Okay, so that was the first dominant belief on these topics. Okay, and so now fast forward to our kind of theories leading up to natural selection to evolution. We have Lamarck, right? Lamarckian theory. And so his big driving theory was the theory of use and disuse, okay? And so this is to say that an organism who uses a trait more often, or finds that there's a trait that would be more advantageous in their environment, they'll use that trait, and then in later generations, that trait will be exaggerated, okay? Or that trait will be passed down to their other organisms. So an example of this would be giraffes. This is usually the common example given with use and disuse, but For example, the early giraffes had shorter necks, and we know this. And so due to Lamarckian theory would state that the physical act of the giraffe pulling its neck or straining its neck to grab the leaves eventually made it so that the giraffes had longer necks. Therefore, the giraffes figured that having a longer neck would be more advantageous in their environment, so they passed that trait to their offspring. To make this even more relatable in humans, this would be like Dwayne "The Rock" Johnson, right? He's really muscular and buff. To say that if he had a child, his child would also be very muscular and buff, right? And we know that's not the case. So this theory has since been excluded. This is not true. But it's important to kind of look at these and to see why things are wrong so you can understand why certain theories are right. Okay? Okay, so that's the Lamarckian theory, the theory of use and disuse. Now let's talk about Charles Lyell. Okay, so Charles Lyell was essentially looking at erosion. He was looking at how erosion could produce our large geographical structures over time. For example, wind, things like wind, water, volcano eruptions. These all produce geographical structures. Okay? And so he also proposed or saw that erosion occurred over a pretty steady, constant time, or at a constant speed. And so if one could determine how much erosion has occurred on certain rocks or in certain areas, then one could also determine the age of Earth, and that's what he did. So Charles Lyell, his important finding was finding the actual age of Earth, which was really important considering it gave us a timeline for evolution, for the origin of from all the way from the first organism to now. So finding the actual age of Earth was very important at this time. So that's really what Charles Lyell did. That's part of what he contributed, was finding the actual age of Earth. Okay? All right. So next, Alfred Wegener. Okay? So Alfred Wegener was looking at species, and he found that there were species on different continents that were related, but they were separated by oceans. And how could that be the case? Well, Alfred Wegener proved the existence of Pangaea. And Pangaea was the theory that all the continents at one time were put together kind of in a puzzle, and that over time this Pangaea broke apart into the continents that we have now. But this is important because it gives an explanation for why organisms that are separated by oceans and natural barriers are related or could be related to each other. Okay, so when you think of Alfred Wegener, think of Pangaea. Okay, now the big dog, Darwin. Let's talk a little bit about Darwin. So Darwin's big claim to fame was the theory of descent with modification. Okay, so descent with modification is saying that one species gives rise to another species over time. Okay, and so this is talking about how we can start with one species and the accumulation of mutations or the accumulation or natural selection really can be the driving force for speciation or for the creation of new species. Now, when we talk about natural selection as being a driving force for evolution, which was proposed by Wallace, right? Wallace is the one scientist who proved or proposed that natural selection is a driving force for speciation. When we think about when descent with modification, we're talking about how natural selection selected for certain traits, and these accumulation of traits could lead to the creation of a new species. Okay? All right. So if you have any other further questions about any of that so far, feel free to shoot me an email or come to the help session. But with that, we are now going to jump into the evidence for evolution. Okay. So first, we should talk about the importance of fossils. Okay. So fossils kind of give us a kind of a sneak peek into the past, right? We're seeing actual structural evidence of the past. And we can also use these to date individuals, right? So through methods we'll talk about in a minute, we're able to date fossils, see at what point in the life of Earth did they come up. We can see what other individuals they may have been related to, what individuals that they may have given rise to that are around now. So the fossils are very helpful and useful for determining what organisms were alive in the past, and also for seeing how they're related to others. Okay. Okay. So what is a tiktaalik? A tiktaalik. So the tiktaalik is one of the organisms that was mentioned in your PowerPoint, and it's the transition between a fish and a land-dwelling animal. Okay, so I would think that that's kind of the important thing to know about tiktaalik. So I would just keep that word in mind. Okay. So now I kind of alluded to fossils. Part of the importance of fossils is being able to date them to determine when those organisms were alive, and also how they relate to others. Okay, and so in order to do that, we need to be able to date fossils. Okay, so there are two main ways that we can date. We can use relative dating or radiometric dating. So relative dating is looking at the position that the layer of rock that the fossil was found in, and if it's known, if the age of the layer of rock that the fossil was found in is known, or if some other fossils that were found near that fossil, if their age is known, then that information is used to find a rough date for the new fossil. Okay, so it's more relative. It's relative to the layer of rock that fossil was found in, but also to other fossils that were found near that fossil. Okay, so it's more of a you're dating based on the other fossils that were found there, or relative to the age of the rock that the fossil was found in. Okay, and this contrasts radiometric dating. So radiometric dating is where we're measuring the amount of isotopes that are present in a fossil, and we're using that to determine the age of that fossil. So over time, radioactive isotopes will break down into another element, and we saw this in carbon dating. And so if we can determine how much of that new element is present, then we can determine how old that fossil is. And so if you really want to think about it, you could say that radiometric dating is a more direct way to date the fossil, because you're looking at the amount of isotopes that are present in that fossil. And that directly contrasts with relative dating, where in relative dating, you're looking at the relative date of the rock layers the fossil was found in, and the other fossils that were found near that new fossil. Okay, so that is a little bit about the dating of fossils. Now let's jump into some more topics. So what are tetrapods? So tetrapods are, gosh, I can't talk right now. Tetrapods are vertebrae with 4 true limbs. Okay. And these limbs, these tetrapods, are known to be jointed, known to have jointy, bony appendages with digits. Okay, so this is just a vocabulary word. I wanted to put it in here just to bring it to your attention. I would say that's maybe a vocabulary word you want to remember for your exam. Okay. Next, homologous structures. So homologous structures are very important, so I'm going to make sure to take enough time to talk about this. So homologous structures are similar structures due to inheritance from the same ancestor. So really we're saying that individuals that have similar structures or similar bodily structures will have similar ancestry, okay? And so to think about this, homologous structures will have a similar physical structure. However, they may not always have a similar function. For example, if you look at a human arm, of course not right now, but if you look at the bone structure of a human arm versus the bone structure of a whale's fin, the bone structure of a dog's paw, and the bone structure of a bat's wing, you would see that we all have digits. We all have a forearm. We all have similar bone structures. However, we don't use them for the same function, right? We have arms. Dogs, well, they walk. Well, we walk too, but you know what I'm saying. Whales swim and bats fly. We all use our appendages for different functions. However, we have a similar underlying bone structure. That is homology. And that is implying that we have a common ancestor at some point down the line. That's why we have a similar bone structure. Okay, so when you think of homologous structure, you know they have a similar structure, not necessarily a similar function always, but possibly. But the big thing is that it implies that there's a common ancestry. Okay, so homologous structures, similar structure, and implies that there's a common ancestry somewhere down the line. Okay? All right. So my last point on this lecture is vestigial structures. So vestigial structures are structures that are inherited from an ancestor that may no longer serve a purpose. For example, in your lecture, the example was snakes with leg buds, right? Embryos of snakes with leg buds. It's obvious that snakes do not use legs. However, the fact that they still have those leg buds implies that they have an ancestor that did have legs, right? And they were involved to eventually not have legs. For example, in humans, we have an appendix. We no longer have a use or a need for the appendix, but the fact that it's there kind of implies a common ancestor where the use of appendix was necessary. Okay, so those are our vestigial structures. All right, so we are chugging along. Only 2 more presentations left, so bear with me. So now we're going to go into theories. Wow, I spelled that wrong. Theories for the origin of life on Earth. Okay. So, one of the worksheet that I uploaded for this presentation has a chart where you can fill in each of these theories and to kind of describe them. So I recommend that you can go in there, grab that worksheet, and fill in the chart. I think that's a little bit helpful for organizing the information in your mind. But we'll just go ahead and jump into this. So the first theory that I want to talk about is the primordial soup theory. And this primordial soup theory is saying that, I'll go ahead and read the definition. So lightning struck the early ocean. The ocean was full of inorganic chemicals. And this lightning strike created the first amino acids, which were the basis for early life. Okay, so this can kind of feel jargony. It might not exactly make sense, but what we're saying is that we have a body of water that has all the necessary components for life. The lightning strike provided the energy to start the process of creating our complex molecules, our amino acids. Okay, so think of the lightning strike as being the catalyst, the energy to create our molecules that are necessary for life. Okay? Okay, so now let's talk about RNA world theory. So RNA world theory is saying that RNA was the first molecule for life. that RNA can self-replicate and build organic molecules. So this, if you remember back to our exam 1 material, when we were talking about transcription translation, we saw very plainly that RNA is really a jack of all trades. It wears a lot of hats and has a lot of roles in the body. A lot of them we haven't even gotten into and that we won't get into. But even just seeing that RNA carries genetic material, it's necessary to make proteins, right? Our RNA make up our ribosome. It's necessary to make our mRNA transcript, right? And in order to make our protein, so that's RNA polymerase, making a messenger RNA strand. So already there, we're seeing how big of a role that RNA plays in the body, and especially with making proteins, the building blocks of our bodies, right? And so it makes sense to think that RNA was one of the first molecules necessary to make life, right? Especially to make complex life, okay? So this theory is really playing into that idea of RNA playing a very strong role in life as we know it now. And so it must have been, according to this theory at least, one of the first established mechanisms in place, okay? All right, so now let's jump into the Clay hypothesis, okay? So the Clay hypothesis says hydrothermal vents in the ocean heat common chemicals to produce amino acids and simple organic molecules. Clay soil collects and protects the new molecules as they get more complex. Okay, so this is saying that, we have our common chemicals and amino acids present. And instead of where the primordial soup theory, there's a lightning strike that produced the energy, we're saying that heat from hydrothermal vents provided the energy in order to create our complex molecules. Okay, so, and we're saying that after those molecules were activated from the heat from the hydrothermal vents, those molecules were then collected into clay soil, and those molecules were protected by the clay soil so that they could become more complex. Okay, so really just try to remember the big basic points, especially ones that were mentioned for each of these theories. Maybe go back to your presentation and read the explanation that was given to you. I would say a big word of advice is to make sure that you can distinguish between the 3 theories. Thankfully, they're all very different. But if you can just kind of explain in broad terms what's being said by each theory in your own words, then you would be good, okay? Okay, so what are some characteristics of early Earth? So really I'm talking about what was the environment like? What were the conditions present in early Earth? Okay, so for example, the atmosphere lacked oxygen, okay? Our early prokaryotes were not oxygen using, okay? They were in anaerobic conditions. And so it's important to note that there was not oxygen or sufficient oxygen present in early Earth, okay? So that could not be utilized as a source of energy or sustenance, okay? So there had to be other chemicals, molecules in place during early Earth for those early organisms, okay? So that's really the big thing I wanted to mention is lack of oxygen, in that the first organisms were unicellular prokaryotes, okay? So with that in mind, what were the first multicellular eukaryotes? And that would be green algae, okay? So just in case it comes up, I wanted to mention that the green algae were the first multicellular eukaryotic organisms. Okay. So now jumping into the Cambrian explosion. So what occurred during the Cambrian explosion? So during this time, animal life increased rapidly and became more diverse. Okay, so that's one of the big things that happened in the Cambrian explosion. Also, you'll see in your PowerPoint the big chart of animals that were present. It's important to know that during this time we had a large amount of diversifying of our animal life. Okay. Well, what about adaptive irradiation? So first off, when does adaptive radiation occur? Well, adaptive irradiation occurs after mass extinction. So a good way to think about this is that after an extinction, the animals or the organisms that were left behind that didn't die off during mass extinction, they diversify. So they themselves diversify and occupy available niches that were vacated from the animals that died during the mass extinction. Okay, and if you remember from class, niches are just another word for jobs or roles in the environment. So the organisms that were left behind after the mass extinction were able to fill those niches that were vacated after the extinction. Okay, so those are some big things to think about with adaptive radiation specifically. And so during what period did the dinosaurs go extinct? That would be the Cretaceous period. Okay, just memorization. So again, the dinosaurs went extinct during the Cretaceous period. Okay, so what is biogeography? Okay, so biogeography is the study of how organisms are distributed in geographical space. Okay, so again, this is important for determining where organisms are, how organisms related, patterns and things like that. So just point definition, important to know that biogeography is the study of how organisms are distributed in geographical space. Okay, where are they? What are their habitats? Where do they live? Things like that. Okay? Okay, so next we want to talk about plate tectonics. So plate tectonics, if you look at Earth's crust, is not one solid sheet, okay? We're broken up. That's why when we have earthquakes and things like that, it's the movement of our plate tectonics, okay? So those plate tectonics are those pieces of the Earth, right? Because since the Earth's crust is not one solid sheet, right? It's broken up, has cracks in it, right? It has character. Those are plate tectonics. And when those plate tectonics move, right, that's when we have shifts, okay? Okay, so what is convergent evolution? So if you remember earlier when we were talking about homologous structures, we were talking about how homologous structures are organisms with similar structures or similar physical appearance in some cases, not all. But they can, those similar structures can imply that there's a similar ancestor somewhere down the line. However, convergent evolution becomes an issue with this thinking, because convergent evolution is saying that there may be organisms that have similar characteristics, but that doesn't mean that it was due to a similar ancestry. It means that they adapted to their environment in similar ways, but independently of each other. For example, let's think about a sugar glider and a flying squirrel. Looking at these, you would think they're very similar. You may even think they're genetically related. They both, right, have that flap so that they can glide in the air. However, these two organisms are not genetically related at all. They don't have a similar ancestor. They both just adapted in the same way to their environment. This is called convergent evolution, and it can cause problems when you're trying to determine the ancestry of organisms or whether organisms are related, okay? So the big takeaway is that convergent evolution is the fact that some organisms that are not closely related can evolve to their independent environments in similar ways, okay? Now let's talk about taxonomy. So taxonomy is a way to classify individuals based on similar characteristics, okay? And this can also help to determine what organisms are in similar family, or what organisms are related or similar. Okay. And so this is a way we kind of classify our individuals. And so this was the classification that Dr. Dickerson asked you to know. So we'll go ahead and talk about that. So I apologize if this is backwards, but hopefully you can still kind of get the big deal, the big gist. So taxonomy should be thought about as a pyramid, okay? As we go up the pyramid, we're getting more specific. But as we go down the pyramid, we're getting more broad, okay? So that's to say, let's go ahead and fill in our sections. So our bottom will be our domain. Next, we'll have kingdom, phylum, class, order, family, genus, and species. Okay, so as we get higher up the pyramid, we're getting more specific. So the top being species, being that organisms who are in the same species are more related than organisms that are in the same kingdom. Okay? And so if I was to tell you, if you know that two organisms are in the same class, would you know that they're in the same family? Not exactly. They could be, but you can't imply that because we're getting more specific. However, if I tell you that two organisms are in the same class, does that mean they're in the same kingdom? Yes, because if class is more specific than kingdom. We're more into a more specific category. So if going back down, we would know that if organisms are in the same class, then they very well must be in the same kingdom. Okay, so I would be sure to remember those relationships, and I will tell you actually now how I have learned to remember these classes. So starting with domain. So domain, kingdom, phylum, class, order, family, genus, species. I like to say, dear King Philip came over for great spaghetti. And kind of silly, but that helps me to remember the categories. Okay? All right, so lastly for this chapter, we're going to talk about phylogenetic trees. Okay, so phylogenetic trees are ways for us to see how closely related some organisms are, for us to be able to track inheritance, well, to be able to track the presence of species. So, for example, you can see how closely related certain organisms or certain species are, and how closely they are to the common ancestor. So you can see some examples of these. You've probably had these in your homework. You've had them in lecture. I won't go over the specifics of those, but if you have any questions about that, please feel free to email me or to see me in the help session. Okay. All right, so let's jump on into our last lecture. This was the one from today about community ecology. Okay, so what is community ecology? So that is the study of interactions between species and communities on varying spatial and temporal scales, also biotic and abiotic interactions. So we're looking at how organisms are interacting with each other and their environment, whether that's living or non-living, okay? Or whether living and non-living aspects of their environment, okay? All right, so let's talk about the different species interactions. So for these species interactions, we're thinking about whether the species is helped, hurt, or not affected. Okay, so for our first interaction, competition, right? Well, this seems pretty obvious, right? But in competition, both of the organisms are hurt. Okay, and so, for example, the example in class about the rams hitting their heads together. They're doing that as a mating practice for being able to have mating rights with the females. And so they are competing for mating rights. However, it's hurting both of them in the idea that they could injure each other or things like that. But another example would be birds. So certain birds are very territorial. Okay, and so for example, if two birds were fighting over access to a nesting site, right, or access to water, access to a food source, they are fighting because materials or resources are scarce. And so they're both already hurt. But them also fighting on top of that is hurting their fitness, hurting their well-being even more. Okay, so in that situation, neither of the organisms ultimately benefit from the interaction. Now let's talk about predation and parasitism. I'll go ahead and lump those into the same thing because in both of those, one organism benefits while the other is harmed. For example, a lion eating a zebra, the lion's benefiting, it's getting resources. However, the zebra is harmed, it's dead, right? So in that example, it's very obvious that one organism is ultimately benefiting while the other is being harmed in the process, okay? And so that's the same thing for parasitism, right? If a dog has a flea, the flea is getting nutrients from the dog. However, the dog is not benefiting from that interaction, right? Okay. Now let's talk about commensalism. Commensalism is where one individual benefits while the other is not affected at all. For example, you see in class, and maybe you've seen Planet Earth or Nat Geo Wild. I know I'm a nerd for those videos, but anyways, birds, right, in Africa or where elephants are present, some birds will ride on the elephants' backs, right, for transportation, but also certain species of birds will eat small bugs that come out of their hiding places when they're disturbed. So riding on the backs of elephants, the elephants, when they stamp down on the earth, it disturbs that area and the bugs fly up, and then the birds can go out and eat those bugs. Now, quite obviously, the bird is benefiting while the elephant is neither helped or harmed. It's just doing its thing, right? Okay. Okay, so now last one is mutualism. Mutualism is where both species benefit. Okay, so a nice example that was mentioned in class is mycorrhizae. Now, mycorrhizae are fungi that grow in association with roots. Okay, so the mycorrhizae is where the mycorrhizae are associated with the roots of trees. So the roots in the mycorrhizae, think of them as long kind of strings, right? Long stringy fungus that's in the ground associated with the roots of the trees. Now, the mycorrhizae increase the surface area that's able to be kind of innervated by the roots and the fungi. So the fungi help the roots to absorb more water and nutrients from the soil, right? Increasing the surface area so they can get more of that soil, more of the nutrients. In return, the plant provides, or the roots provide, the mycorrhizae with some sugars for nutrients, okay? So in a way, they both help each other. They both benefit from the interaction. Okay, so that's really the important point. When you're faced with questions concerning the relationship between organisms, you want to think about, is one of them, are there some organisms that are benefiting? Are some being harmed? Is one not being harmed or helped? That will really help you to break that down and to answer those questions right every time. All right, so now let's talk about keystone species. Keystone species are those very important organisms that help maintain the integrity of the environment of that habitat. For example, starfish. So starfish, you probably didn't know this. I didn't know this until I came to college. But starfish are fierce predators, okay? And starfish they are capable of eating or preying on some of the main other predators in their environment. Of course, not sharks, but like some other small predators in their environment. So essentially, they're keeping the number of individuals down or in the environment or at a certain level so that no one of the other organisms dominates the environment. Okay, so the starfish helps to keep the other organisms' numbers in check. Okay, so if, for example, the starfish was no longer present, then some of those other organisms would increase in number, right? And some organisms would go extinct or die off. So essentially, starfish help to keep these other organisms in the environment in check, okay? So that's just one example of a keystone species. I know also that wolves are keystone species. Those are the only ones I can think at the moment. But I think in the PowerPoint that's uploaded on Blackboard, there are some other examples that you can see there as well. Okay, so let's also think about some invasive species. So invasive species are species that are present that were not native to that environment. For example, we talked about bluegrass for Kentucky, kitties. Domestic cats are an invasive species. They're very good predators and they can cause problems. They kill a lot of birds, apparently. Also, some other types of plants as well can be invasive species. So essentially what we're saying is that these invasive species are so damaging because there's no predation cycle set in place to keep their numbers down. So essentially these organisms can just grow and take nutrients and develop unchecked in that environment, which can cause problems for other organisms that are native to that environment who don't have safety guards set in place to protect themselves from that species or other species organisms that will prey on them and keep their numbers down. Okay. Okay, so let's think about climate change and global warming. So climate change, think about that change being a smaller scale, changing the precipitation patterns, severe weather patterns, and other biological systems within an environment. And global warming is a specific example of a change, right? So global warming is talking about how the increased concentration of carbon dioxide in the environment has actually kind of created a thermal blanket around the Earth. So before our CO2 levels were so high and other greenhouse gases, of course, when the sun rays hit the Earth, some of the rays were able to be bounced back and reflected back into space, right? So this kept the Earth from getting too warm. But over time, as the concentration of CO2 and other greenhouse gases in the atmosphere accumulated, it kind of formed a blanket, if you will, around the Earth. And this keeps some of the sun rays or some of the heat from being able to bounce back into outer space. So therefore, that heat is being kept in the Earth, right? Or kept reflecting in the Earth, okay? And being absorbed by the Earth's surface, if you will, okay? So with global warming is troubling because we'll see an increased rate of ice melting, altered ocean currents. It can take a toll on many aspects in our environment other than just the temperature, okay? So that's important to note. So we already talked about the increased level of CO2 in the atmosphere being concerning with the relative, with concern with global warming, okay? And so last, what is phenology? Okay, so phenology is the study of the timing of biological events in plants and animals, okay? And so what is their internal clock? That's what we're talking about. So, for example, how do bears know, or Actually, bears don't technically hibernate, but what animals that hibernate, how do they know when it's time to hibernate? How do geese know when it's time to fly, to move for the winter? How do flowers know when to start opening their petals? They all have an internal clock, an internal mechanism for determining when it's time to do certain events. Okay? All right. So those are that, and I am done. So that is all of the outline. Those are some of the main topics talked about on this exam. I'm not. So that'll be all that I go over in this video. If you have any other questions about the material, feel free to shoot me an email or to come to the help session tomorrow from 5 to 5:50 in Davidson 209A. Also, I'd like to say that I have included some helpful videos at the bottom of this outline that go over some of the more difficult concepts covered on the exam. So if you have any trouble with some of these topics, some of these videos may be able to help you. Also, so this video will be up on Tuesday, and also all of the worksheets and everything. So all of the material has been uploaded. Also, I will flash this one more time just in case you are not able to sign in. So if you were not able to sign in, please go ahead and do so. All right. So that is all I have for you. Thank you for tuning in. Good luck on your exam.